And so our next speaker is Dr. Grace Brannigan from Rutgers Camden Department of Physics. And her lab is interested in computational biophysics and molecular simulations, and has a particular interest in ion channels. And she's going to tell us about, you guessed it, an ion channel, which is the E protein of SARS-CoV-2. Grace, over to you. Oh, you're muted, I'm sorry.
So I was asking if you guys could see my screen, but I guess I needed to ask first if you could hear me. Can you hear me now?
All good on both counts, Grace. Thank you.
Okay, great. Okay, so thank you, Sagar and Stephen, for inviting me to give the final talk in this great crash course. So I'm going to talk about the SARS-CoV-2 envelope viroporin, and let's see here. The outline of the talk, I'll give some just basic information, the role in the virus life cycle. For the E protein, there is really not that much structural biology information that we have, which is a little bit surprising because it is a small protein. It seems like it should be relatively straightforward, but I can tell you that as of the last week or so, the number of structures that we have for the full pentamer has doubled. Because we have an additional one. So I'm going to give you some information about what we do know, the conveniences and the significance, why we would like to know more about the E protein and the challenges that people have faced. And then just talk a little bit more about the available structures and what they have in common, what are some major differences. Then I'll talk some about target sites, and the most obvious and highly sought after target site for the E protein and for any ion channel that you want to block is the pore, but there's other potential sites here as well, which are particularly useful because the E protein is more than just an ion channel. Give some information about the evolution during the pandemic, and finish up. So the basics of the E protein. This guy is a 75 residue globular transmembrane protein. It is a homopentamer, so it has to be a pentamer because the center of the pentamer is where the pore is. So just checking, you guys can see my mouse?
Yes, we can.
Okay, thank you. So the pore is at the center, and we have 5 subunits around. So if you don't have a pentamer, or you don't have an oligomer, you don't have a pore. And if it's smaller than a pentamer, the pore will be way too small for any ion to go through. So so you have to have this oligomeric structure. It's co-translationally inserted into the ER, transported to the Golgi. Despite its name, most of the E protein is actually not localized in the viral envelope. So most of it ends up sticking around in the ER and in the interface. And so what its role there is not really understood. We do know that it does play a role in assembly and membrane curvature and viral shape that is not directly tied, as far as we know, to the fact that it's an ion channel. So so this is a figure from the very recent structure that was published. And so, certainly initially we have the virus, it binds to ACE2, and you have the RNA replication. The E protein here is represented by this little red guy. And then once you have the assembly and budding phase, some of the E protein does stick around in the Golgi and the ER, but there is this assembly process where the E protein is required. So if you delete the E protein, instead of getting our classic coronavirus shape, you get these long, stringy, amorphous viruses. So it's not clear why that should have anything to do with the ion channel role of the protein. It seems more like it has to do with the interactions of the E protein with either other proteins or the membrane itself. But the M protein alone is not enough to determine the viral shape. If you delete the E protein and 3A, you lose viability. Just deleting the E protein on its own, you lose virulence. But directly mapping these effects to the ion channel conduction properties has been challenging. So far as I know, the only direct connection that's been made is that if you are able to block transport of calcium through the E protein, then this inhibits inflammation. So there are a couple known blockers of the E protein. One is amantadine, which also blocks the M2 channel in flu. Another one is amyloid derivatives. There are many others that are under investigation, but as I said, we've had a real shortage of structures. So because the E protein has this dual role, where it's both an ion channel and it affects the membrane shape and the process of budding and the envelope shape and everything. It's a pretty desirable target. It happens that only 4 residues differ between SARS-CoV-1 and SARS-CoV-2. So over here on the right, I have an alignment, and you don't have to know the details of these alignments, but basically what you can see is that everything is purple for the most part, except for a few stripes of white here. And so there's just 4 residues that differ. And so that means that a lot of the research that was already done on SARS-CoV-1, and indeed the first structure that we had was for SARS-CoV-1, is really seems to be directly applicable to SARS-CoV-2 as well. So this protein has a pore lining helix that's shown here in silver and a cytoplasmic facing C-terminus. So that's shown here in green. And what we find is that we know that the pore lining helix has this sort of interesting relationship with the membrane, where the pore lining helix on its own is about the right size for the membrane, kind of, but then we have this green, what's shown in green, this terminal helix, and that terminal helix ends up causing a bunch of questions and problems, and how it interacts with the membrane is unclear. And I've tried to draw lines right here to show where you would expect the membrane to lie, but you will see a little bit farther in the talk, it's not a very straightforward question. The function and the conformation of the channel are both really sensitive to the lipid environment. So it's known that the selectivity of the ion channel can be removed if you remove anionic lipids from the membrane. The multiple structures that we have, i.e. the 2, are done in different lipid environments, and even within, we know from the researchers who determined those structures that they were unable to resolve certain regions of the proteins, and depending on the lipid environment that they used. There is also post-translational additions of palmitic acid to these membrane proximal cysteine residues, which are up here. And so those guys may also affect interactions with the M protein, as far as there's some evidence that shows that for SARS-CoV-1, and those are not included in structures. So the 2 structures that we have. So on the left is the shiny new structure that just came out from the Hong Lab, and it is a solid state NMR structure. It's for SARS-CoV-2. It's only residues 8 through 38. So I said that this was a 75 residue protein, and the older structure that we had has residues 8 through 65. This one is only residues 8 through 38, but it is in a more realistic membrane mixture. So it includes POPC, POPI, POPE, POPS. So we have a mixture of charged lipids and neutral lipids. It's got cholesterol in it, and it's 2.1 angstrom. So it's much higher resolution than the previous structure. The pore is actually usable potentially for drug design. It is missing this, the funky green terminal helices that I mentioned cause problems. And it is not just that they are unresolved, it is that they are not present in the construct. So this is a figure from the authors of the new structure comparing the two available structures. And on the left is the new one, and on the right is the previous one. And so the pore in the older one is certainly more open. It has got this twist to it. The new one is very kind of ordered and structured. As I'll show you in, I think, the next slide, it also happens to be closed. But they're done in different lipid mixtures. So actually the one on the right was done in LMPG micelles. So this was a solution state structure. but they also have a different amount of the sequence. So basically, the one on the left has only about half as much of the sequence as the one on the right, a little over half. Okay, so that being said, let's have a look at the higher resolution structure, the shiny new structure that we are really happy to have. So, so these are several different representations of this structure. And so like I said, it's an NMR structure. And so for with NMR, usually you would generate multiple models that all satisfy the NOE constraints. And in this figure right here, we have these models overlaid on top of each other. So you can see that for the pore, with the exception of the, the two ends, there's really a lot of agreement. The constraints that they have are, the model is not underdetermined by the constraints that they have available. Then next to this one, we have the structure with a pore, space filling model of the pore. And so this silver outline shows basically how big is the empty space in the pore, how wide is the pore. And there's this region right here where the pore is really, really narrow, which means that any ion that is, that is trying to pass through the pore has to go through this very tight constriction. And in general, these constrictions are hydrophobic as well. So the ion will have to go through the hydrophobic constriction or hydrophobic gate, as we sometimes will call it in ion channels, without any water around it because it's so small. And then it was also interacting with hydrophobic residues, which is going to be pretty unfavorable. So, so farther over here on the right, we have actually the radius, the pore radius. And again, there's this very tight constriction at L18, where the radius of the pore is less than 2 angstroms and actually is even less than 1.8 angstroms. So getting ions through there, even without water, is going to be very hard. So even the authors of this paper say that apparently this does seem to be a closed structure. And it's interesting because the E protein is not a viral protein that we know about it as being gated. And so the existence of a closed state was not assumed beforehand, a priori. And it might be natural to think that this structure is therefore not going to be that useful for the design of blockers. But one of the things that we know from the design of blockers for other ion channels, and indeed for anesthetics where we would prefer they don't block ion channels, and sometimes they do, is that it's actually pretty common for blockers to have a high affinity for the closed state. And this is fine. It's actually good, because what happens then is that in a kind of negative allosteric modulation, it destabilizes the open conformation. And so it's a little bit weird to talk about allosteric modulation with a pore blocker, because you certainly couldn't have a positively modulating pore blocker. But if the blocker has a higher affinity for the closed state, then you will still have a reduction in the open population of the protein, which is ultimately what we want. So the existence of this closed conformation, even if it is found only under certain artificial conditions, is still very, very useful information. Okay. So these are NMR structures. They were not able to resolve interactions, orientations or conformations or binding modes of individual ligands, but they were able to determine the structures again in the presence of known blockers. And so, like I said, the most well-established blockers are amantadine, which is the M2 blocker, and the flu channel, and then this guy HMA. And so they both have affinities in about the 10 micromolar range. And so what they can do is they're looking here at the chemical shifts, how the chemical shifts change upon binding. And certainly we already know that the end parts of the protein are the ones where the structure is the least well determined. But they do see that when you have block by these molecules, you do have significant changes in the chemical shifts on the two ends, also at S16. So that does lead them to believe that the drugs most likely bind on this end of the channel. And so what they were able to do then is they said, okay, we're just going to dock the drugs to the structure. And so this is HMA docked to their structure. Of course, with docking, there's a lot of different issues with docking, and one of them is that it can be really challenging to tell which of many possible modes is the right one. And certainly the SI, even for this paper, has a lot of other different modes that you can dock HMA in. That doesn't mean that they aren't. It is very possible that HMA has multiple conformations in this pore. It is more likely that one of them has a particularly high affinity, and we need to figure out what that is, potentially using computation. But unlike many of the other structures and complexes that have even been presented today, we do not have. I cannot tell you, okay, these are the direct interactions that stabilize HMA in the E protein pore. I really don't have any idea beyond what is shown here, which is that there are stronger chemical shifts, in this region rather than that region. And so there is just a lot of work here to be done. This is a really kind of the Wild West so far. So I had talked about the pore sites. There are also some possible non-pore sites. And one thing I did want to mention is that blocking the E protein pore does introduce, if we target that pore, there is a potential challenge that is introduced, which is, so one of the things is that the E protein does more than just act as an ion channel, so you're not removing that function necessarily. But the second one is that we have, humans and other eukaryotes have pentameric channels that are really important for, for health of humans, and we don't want to block those. And so whatever blocker that can be designed, it has to be for the clinical concentration to be less than the toxic concentration. It has to be highly selective for the pores of the virus and not for the somewhat similar human pores. And so for that reason, it's also really nice to look at potential non-pore sites. And so my student, Jesse, ran some MD simulations, and I'll show you some movies. I put all the movies on one slide in case they don't play. But he ran simulations of this E protein. This is the old structure, and these are coarse grain simulations in a membrane of sort of normal thickness. So these are, each lipid has hydrocarbon chains of about 16 or 18. And what he found in these coarse grain simulations was he observed that the membrane actually spontaneously bent within the simulation. And we hypothesized that that might be a result of the fact that the protein is thinner than the membrane and that it has this asymmetry introduced by the cap. And so as a reference, the normal kind of mechanism that we would expect here is for symmetric hydrophobic mismatch. So this is a hydrophobic mismatch mechanism, is that you would have in the ideal case, the protein, which is represented by the green blob here, matches the membrane. And if that doesn't happen, if it's too short, you get this kind of bow tie look to the membrane. The membrane actually deforms. And this is also sometimes called the mattress model. But for our case, for this particular protein, because it has this cap on it, that's not an option. It can't get buried into the membrane because this cap actually does have a lot of charge residues on it. And so instead, the cap is forced to stay above the membrane, and the rest of the membrane bends around it. And so there was a pretty straightforward test of this hypothesis, which was just to see how the membrane shape changed with different membrane thicknesses. And indeed, the shorter you make the membrane, the less mismatch you introduce, the less bending we get. So one of the things that suggested to us is that these effects on membrane shape are determined by the lipid protein interface. And so if you had drugs that bound to the lipid protein interface and basically mediated that interface so that you did not get these deformations, that the whole assembly and budding process that the E protein is really key for would be interrupted. Also, like I said, the pentamers you can't have an ion channel without the pentamers, and probably whatever effects you get on shape, you can't have either. And so potentially binding to the subunit interface between helices could be useful as well. If we could destabilize these pentamers, then that would be fantastic. So I think that people do need to think beyond the channel as a potential target, but we are still really excited to have the new pore, high resolution pore structure that we have. So I said I'd have movies. I put them all on one slide. This one is a coarse-grained MD simulation of anionic lipid binding. And so the anionic lipids are red here. And I'm just going to play this. Basically, what you see is that they end up concentrating in certain regions of the protein. And so if we were able to do to get cryo-EM structures of this protein, then potentially we could actually have specifically bound lipids and how they interact with the protein shown in the structure. We don't have that. So we're not reduced, but so we need to use coarse-grained simulations to figure out where lipids bind. This is I'm just going to pull this through, a movie of the membrane bending. Very early on it happens. You can see if you place the protein right here so that this charged cap is not buried in the membrane because it shouldn't be, then what happens is the membrane bends up around it. And then finally, this is our first atomic simulation of the first structure, and goes to show why we say we needed better structures, and we're really happy to have a new higher resolution structure. It was definitely not stable in the atomistic simulation with just plain old POPC lipids. So now I am going to move on. Okay, wait. You guys get to see the movies again. I always tell my students to play movies twice anyway, so there we go. And okay. So now I'm just going to finish up by talking about the evolution of the E protein. And so the same project that was already discussed for many of the other proteins, that team also investigated the E protein. And so what they found was that the E protein is genetically quite stable. Of their sample, almost 99% had unchanged E proteins. There were 81 unique sequence variants, 79 were single mutant variants, and there were 3 doubles. There were about half and half conserved and non-conservative. Most of the non-conservative changes lied in the boundary and surface portion of the protein. And the slight majority, 66% of them, seem to destabilize the protein. But I want to emphasize here that when you're talking about destabilizing mutations for an ion channel, that does not mean that it will lower the function of the channel, particularly because for an ion channel, stabilizing the protein can mean closing the channel. The closed state is the tightest, most compact state, and that is not a functional state. And so if you introduce a mutation that actually causes a little bit of repulsion between the subunits, you can open up the channel, and then you actually have a more functional channel. So in this particular case, destabilizing the closed state for the open state would be a sign of a more functional channel. Some notable mutations that they found, serine 68 phenylalanine. So this guy is outside of even the longer structure we have. It's in that terminal region. My guess is that this one could potentially stabilize the interactions of the terminus with the membrane, because we know that aromatic residues have a high affinity for the interface, the hydrophilic interface of the membrane. And so this mutation is going to be kind of far away from the pore in that part that needs to stay close to the membrane. Then there were also these mutations of leucine 37. And so leucine 37 is right at the end here of this is the high resolution, just pore construct. And so the cap, if we had it here, the long green cap would be like out on this side. And this one, L37, for one thing, it was one of the residues that was shown to have a chemical shift change upon binding of HMA. So it is certainly possible that this mutation could influence interactions with blockers. The mutations that they found were mutations to histidine or to arginine. And from my perspective, this certainly would concern me because, again, when you have both of these mutations were destabilizing, but when you have arginines in a symmetric channel, they can repel each other, and then they can actually cause the channel to open up a little bit. So finally, some key lessons. from what little structural information that we have so far, which means that all of it is more valuable, each piece. So we have multiple structures now that confirm the pore lining residues and that it's a pentamer, that we have pentameric stoichiometry. The E protein conformation and function is extremely lipid sensitive, but this is part of why more structures are not available. And so a common approach for getting NMR in of membrane proteins, especially really small membrane proteins, is design of lipid bicelles. And these are, using solution state NMR with lipid bicelles is somewhat controversial. Regardless, they were not able to get this to work in the earlier structure that I presented, and so they were only able to use micelles. The new 2.1 angstrom structure of the pore is really promising for the design of higher affinity pore blockers. Resolving the C-terminus is particularly challenging, but I think it could yield great rewards if not we, we're not experimentalists, but if some groups can do it, because it may be particularly important for the interactions with other proteins and with the membrane. And the E protein is relatively stable genetically, but the mutations do include residues that interact with current blockers and also interact with the membrane itself, or could affect how open the pore is. So with that, I have just a few acknowledgements for this particular work, since most of it was not ours, but the E protein simulations that you saw here were done by my student, Jesse Sandberg. And then a lot of the insights that we had were determined through a long history of working with other Pentameric channels, which I've had several students working on. And then the Office of Advanced Research Computing gave us a lot of hours for simulations here, which we really appreciate. So with that, I will say thank you again and take any questions.
All right, great. That was a great point, Grace, about mutations might be destabilizing, but might lead to more function in terms of opening the channel. I think that's something.
Yeah, I guess, with any protein where you have multiple conformations,
yeah,
you have to be careful about which conformation, you're stabilizing it in favor of what. Yeah,
exactly. I think we have to, comprehensively examine all the states that are available, and then evaluate the impact on all states to really say, with high confidence what's going on. But okay, so the questions, Ricky Cohen asks, since this is an ion channel, what role can this use to mediate processes needed by the virus? Do you think overexpressing in neurons can help show function?
So I think that one of the mysteries actually is still, how does this virus use the E protein? I assume that maybe the question about neurons is because neurons are rich in ion channels, but actually many ion channels are not studied in neurons because they're harder to work with. So I think that we certainly know that, and I can see the second question also is the E protein responsible for only calcium transport, or can it transport other ions? So it can transport other ions. It can actually, it can transport anions or cations depending on the lipid environment. So that in and of itself makes it a kind of mystery, because we know that under certain circumstances, it will mainly transport calcium, but we don't know why. We know that if you block it, it affects inflammation in the host, but it's not clear how it affects the viral life cycle otherwise. And so, I think that we know that we know on at the pentameric level how it works, and yet why conduction is important for the virus is something we don't know. And so I think that those studies would need to be done within the virus. They couldn't be done in another cell type, would be my guess.
I have a related question, which is that E protein has a low copy number in the virus, and why do you think that is? And do you know how that is kind of regulated?
I have no idea. I don't know if other people know. I don't know. I think, yeah, I don't know if there's someone else here who has a better sense of that than me, but I'm not sure. I don't know. Yeah.
Yeah, no, I mean, it's a mystery. Do we know, Victor asks, do we know what is the lipid composition of CoV-2 membrane?
Yeah, so the CoV-2 membrane will generally, so the envelope membrane will take on generally the lipid composition of whatever membrane it kind of burst out of. So the quasi-native membrane that people have referred to is a membrane that that represents, the ER Golgi interfacial compartment. And we have a general idea of what that looks like. So we know what lipids are there. The percentages of what's there, we don't really know. So I think right now there's not information that it is particularly different from your kind of classic mammalian membrane. So neuronal membranes, for instance, look very, very different from most other membranes in the human body or human tissues or mammalian tissues. But this membrane seems to be pretty standard. So they have a sense of the head groups, PC, PE, PS, PI. How the chains, I think one thing that's pretty interesting is that given the effect on shape, actually the flexibility of the acyl chains and their length and the thickness of the membrane will be pretty important. And we just don't have any information that it's any different from the average for this particular membrane.
Stephen asks, Grace, do you know the copy number data for E protein versus M protein in the virion?
I don't. I'm sorry. I'm not the best person to ask about this. So let's see. So I can tell you, I could look it up basically for you right after the talk.
Sure.
Yeah. So
Greg asks, when is this channel active, during budding or entry? Does the deformed shape in the absence of the E protein affect viral entry?
It is not active during entry, or if it is, we don't know that it's active. There's no processes that are essential for entry that have been shown to be, to be really mitigated by the E protein. Does the deformed shape in the absence of the E protein affect viral entry? So I think, as far as I know, what it ends up affecting is actually budding. So I don't know about any implications for entry. I think the shape gets so deformed that the whole life cycle just kind of, the assumption, I think, or the interpretation is that the life cycle is basically stopped at that point once it has that shape. Or is really mitigated and becomes unusual. But yeah, I don't know more information about that. I think that they have the biggest effect that they can see when you remove the E protein is that you lose virulence, and so there is still the virus is not gone, but its ability to reproduce and infect is really reduced, and so. If you reproduce, delete E protein and other proteins, then you have an even stronger effect and you don't have viability anymore. And so at least some of the role of the E protein is through its interactions with other proteins as well.
Mm-hmm. Okay, and last question from Sangyuk is, to my understanding, CoV-2 doesn't seem to have capsid proteins. How then is the osmotic pressure regulated? Is the E protein responsible for this function?
I'm not sure if I totally understand the question. Which osmotic pressure do you mean? Do you mean within the virus, within the envelope, or can you clarify?
Sangyuk, there is a lipid.
Sangyuk, why don't you unmute?
Okay.
Yes, I meant inside the virus.
Okay, so there is an envelope, and so the osmotic pressure across the envelope can be regulated via the viroporin. The E protein can certainly contribute to that. It can in addition to transporting ions, it can also transport water. So the envelope is not sealed. I don't know if that answers your question. I think it's
It
not.
sounds like it would equilibrate, Grace, if there were
Yeah.
changes in osmotic pressure. Yeah.
So I don't think that is there information, maybe I'm not aware, that the osmotic pressure, that there should be an osmotic pressure difference or across the envelope?
I don't think so, Grace.
Okay, yeah.
It's a minuscule compartment. And if there's free flow of water back and forth and some flow of ions back and forth, I would guess that's probably enough to protect the lipid bag from exploding.
Yeah. So there's definitely, I mean, that's one thing that E protein certainly does, is that it contributes to it's a porin, right? It lets anything that's smaller than, a few angstroms go through, and which includes water. So, yeah.
Okay, well, that's wonderful. Thank you, Grace.
You're very welcome. Thank you very much for inviting me.
Show our appreciation again. Thank you.